Well, I'd like to welcome Mike Farrell to the Resilient Biome podcast. And Mike is a researcher in microbiology with a PhD, decades of research in the private sector, working for the government, and now more recently in academia. And Mike, thanks for being with us today.
Hey, Rich, it's my pleasure. Thank you for inviting me.
Yeah, it's sort of an interesting serendipitous story, the way we met. You are the brother of a good friend of mine for the last 20 years or so. And it was a Christmas Eve service and just happened to meet you afterwards. And I think I'd only done my first podcast and I knew from your brother, Tim, that you'd worked for the CDC and doing some interesting work. So being the little science nerd that I was, we had a really fun conversation. I invited you afterwards to consider being on the podcast. So really glad that worked out.
Yeah. I apologize for any for burdening you with my brother. Anything he may have said or done. Yeah, Tim's a great guy, and I'm so proud of him and proud to be his brother. And him and his family are just fantastic.
For sure. Yeah.
I love visiting them.
I end up apologizing for Tim all the time as well. So I get it.
Yeah, yeah.
Yeah. I can see the not so much the physical resemblance, but you strike me as sort of this laid back, fun guy, so I don't know if that you would agree, but I see the similarity.
Well, yeah, I see that in Tim as well, and if you see that in me, I appreciate that.
Yeah, great. Well, one of your background here is I saw you worked for the CDC and you were the director of bioterrorism and rapid response. And my first thought was that sounds like a Tom Clancy novel. With pretty exciting, like you're ready to fly to the latest bioterrorism. So, am I over romanticizing that?
Yes, yes, you certainly are. No, this is a lab set up by the CDC. Uh, post anthrax, uh, the anthrax scare from many years back.
Sure.
Uh, Law enforcement, public health. They come across these white powder substances.
Mm.
Here and there. They get sent in the mail as pranks or uh, etc. And so the CDC, through the public health system, a lot of state public health labs, and in cooperation with maybe the State Department to pull in some, embassy capability, uh, just a screening method for white powders to uh, quickly determine if they're a threat or not. So, the CDC developed a suite of assays and techniques that can be used to interrogate these white powders and rule in and rule out in any threat that might be associated with them. And it happens very frequently, but
Mm.
of course, the vast majority are talcum powder,
Mm.
etc.
Mhm.
Flour. Not meant to be threatening, just non-intentionally dropped or picked up somewhere.
Mm.
But law enforcement's always on the alert for these things. First responders too, and so there's a method that they can use to deliver those to public health and get them screened rapidly. So everything can go back to normal because businesses are shut down, transportation hubs can be shut down. So the more rapidly you can resolve that uh, issue, the better it is for everybody. So that's the purpose of that capability. And the lab at the CDC that I directed was sort of developed the assays, troubleshot, trained, this sort of thing. That network of capabilities that's out there.
Yeah, interesting. So not
Public health.
a Tom Clancy novel, but really important work, especially duplicating that and getting in real time. That sounds really cool.
Yeah, it is important, and thank goodness that we haven't had another incident like the anthrax attacks since then. But uh, preparedness is one of the CDC's strong points, and they strive to do that as best they can.
Yeah, almost surprising, frankly. I mean, bad guys are bad guys and, they create evil. So thankfully, I agree with you on that. Yeah.
Yeah, it's not just the CDC. The Department of Homeland Security, the Federal Bureau of Investigation. There's a whole network of uh, uh, capabilities within the U.S. government uh, and the state governments for combating bioterrorism. Uh, should that threat occur.
Yeah. Well, the average listener is no doubt thankful for that intervention. How did you first get interested in the field of microbiology? And as you talk about that, just give us some more of your background, if you would, please.
Sure. I I attempted to go to college right out of high school, um, but that didn't work out so well for me. I was very immature and interested in many other things, not academic. Uh, so I joined the Navy at a young age, uh, and I did that for 9 years, and got out of the Navy and went back to college, University of Wisconsin, Madison, and got a undergraduate degree in biochemistry. And during that undergraduate degree, I worked for as an undergraduate researcher. I worked for uh, a professor in the biochemistry department named Jack Gorski, and Dr. Gorski, his main interest was estrogen. Uh, so he studied the estrogen receptor and the genes that are regulated by estradiol, the hormone that binds to the estrogen receptor. Hormones are extremely powerful, and they can have dramatic effects on your body uh, in a regulatory fashion, just very minute amounts.
Hmm.
And so I came to appreciate how powerful hormones are. And during then coincidentally during my undergraduate education, I took a class on viruses that was taught by Dr. Roland Ruckert, uh, and that's when I really started getting interested in microbiology. Was viruses. Viruses they have very small genomes, so in order to do what they do, they really have to hijack and take over the host cell mechanism. And coincidentally, one of the ways that they regulate themselves are they do that by using host regulatory mechanisms, and the hormone regulatory mechanisms happens to be one of those that they will use.
Hmm.
So, working in a lab that studies hormones, getting interested in viruses, and find out that their genes are also regulated by hormones because they don't code for their own regulatory proteins. So they use the host regulatory system. And I just thought that was fascinating. And then when I applied to graduate school, I targeted a lab. I did my graduate work at the McCardle Laboratory for Cancer Research, also at the University of Wisconsin Madison. And I wanted to join the lab of Dr. Janet Mertz because she was actually studying hormone response elements that existed in viral genomes and studying how those hormones affected uh, the regulate the how the virus replicated itself. And so, just marrying those two interests from my undergraduate into my graduate degree was, just seemed quite natural. Uh, so, pretty straightforward evolution of ideas coming together in a graduate work.
Yeah, yeah, it worked out well for you. Not that I know a lot about bacteria, but compared to a virus, to me, a virus is like a black box, I don't quite understand it. And I remember reading a book on the Spanish flu years ago, for our listeners, 1918, 1919, terrible outbreak. And being shocked to learn they had not yet discovered even what a virus was. So they're seeing some bacteria there, but I found that to be fascinating. But I'm really at a loss to understand viruses, so that's great.
They're fascinating. I could go on and on about viruses at the drop of a hat. but yeah, the influenza virus has been around, been with us for a very long time, and it's a constant battle to keep that at bay.
Mm. Another reason, I mean, just that illustration, not even knowing what a virus is or that it exists, let alone how to work with it or deal with it. So how important just research in general is, obviously.
Yes, absolutely. The, the 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 frightening thing about the 1918 influenza pandemic is how rapidly it spread. You know, Europe, America, um, and they didn't have even 1% of the kind of globalization that we have today.
Mm.
You can only imagine if an influenza strain like that arose again, how devastating that could be.
Mm-hmm. Yeah. Yeah, that was during World War I, so that certainly helped spread some, but yeah, you're right. It'd be much worse, much worse. Yeah. During my I had undergrad in medical technology, so worked in hospital labs. That was almost 40 years ago now. I had a rotation in microbiology, so that's about as much as I know officially about it, other than my own sort of interest in the last several years. But this was old school, which you're very, very familiar with. For our listeners, old school plating of microbes with your little wand on the agar plates, etc. And I swear, if you put E. coli or Pseudomonas aeruginosa under my nose, I would still know its smell. They would, I don't know if they still do, but they would have us lightly smell it. So obviously, in the last 40 years, there's been many changes in technology. So I wonder if you could just talk us through that and how those technological advances impact your work.
Sure. So many of the techniques you described still go on today. Plating, you mentioned, I think you mentioned in a note to me that, putting antimicrobial discs on a culture plate, that work is still being done today. Very
Interesting.
classic, medical microbiology techniques. Um, in the last 10 years, there has been a uh, uh, huge progress has been made in technology, obviously. We can do whole genome sequencing now. Mass spectrometry also plays a big role in identifying bacteria now. Very high throughput, not one sample at a time, but many samples in parallel. And so that those technologies are really taking over the field. But a lot of the techniques you describe are still being used. That rate of change is governed by a lot of things. A couple of them being, proving clinical efficacy, going through clinical trials and proving that a diagnostic test does what it's supposed to do with, limited false positives and false negatives. Um, Insurance reimbursement is also a big role in that. These new techniques, although they can do a lot of samples at a time, they're expensive. The equipment is expensive. So there's costs associated with changing technologies that also have to be taken into account. But the landscape is definitely changing from what you described. But you would be surprised how many of those techniques that you're used to are still being used today. Um,
Mhm. Yeah, and some of
I,
your,
I,
go ahead.
well, I would just say that I'm not a clinical microbiologist by any stretch of the imagination. I do research, so I use these tools, but not to develop diagnostics. But I don't diagnose clinical samples myself. I just want to make that clear.
Sure, sure. In some of your notes to me, you compared the advances to sepsis, blood sepsis, and that the really state of the art is still blood culturing that takes up to 72 hours. You draw blood, put it in little culture tubes, and then see what it grows. And that struck me that, and I used to draw those blood cultures almost 40 years ago, that this is still state of the art.
Yes, yes, and it's a huge problem. Um, Blood culture is the gold standard still. That can't possibly last much longer. There are so many efforts going on to develop much more rapid methods to diagnose sepsis, but it's a race against time with sepsis. Sepsis is the number one killer in hospitals.
Hmm.
It costs billions of dollars to the healthcare industry every year. The most important thing for sepsis is to specifically identify the bacteria so that you can give the very specific antibiotic, right? So many of these strains are resistant to antibiotic A, B, or C, but not D. So initially they'll treat with a broad spectrum antibiotic when they suspect sepsis, and wait to get the detailed information about the pathogen, the bacteria responsible, so they can fine tune the treatment. And as you said, it takes anywhere from 24 to 72 hours to get that result, and sometimes the bacteria don't do what you want them to do. Meanwhile, the patient is still septic. So, yeah, one of the other projects that I'm working on is really trying to shortcut the diagnosis of sepsis, not by trying to identify the bacteria, but really to detect the change in the immune system, right? The sepsis is really, it's your immune system that kills you in sepsis. It's a over response to a bacteria infection of the blood that results in this out of control cytokine storm of inflammatory and pro-inflammatory and anti-inflammatory molecules fighting against each other, and ultimately your organs just shut down. So race against time to apply the very correct antibiotic, and the sooner you can start that process, the better. I mean, generally you wouldn't even start testing for sepsis until the patient starts showing symptoms. So how do you even start before that? If you could develop a way to detect the upregulation of these cytokines early before symptoms occur, yeah, you could start that process earlier and provide treatment earlier and just have better patient outcomes and save lives. So maybe the key to improving sepsis outcomes is diagnosing the immune response rather than the specific bacteria up front.
And I should have put that together before. Never put that together, that sepsis is fundamentally a cytokine storm, which of course we've heard a lot about that in regard to COVID. So the solution that you're searching for there, would it not also apply to COVID, influenza, et cetera, potentially?
Potentially. It's hard to compare a bacterial infection and sepsis and and a virus infection, because the virus, once you catch a virus, there's 3, 5, 7 days of incubation before you develop symptoms. That's a very small window to think that somebody has caught a virus and to catch the immune response before symptoms show up. You'd have to be pretty fast.
So yeah, so you're trying to mitigate before the cytokine storm starts would be ideal.
Well, ultimately, in the case of sepsis, you're not really mitigating. The way you're mitigating the cytokine storm is you're killing the bacteria that's causing that.
Okay.
Right. So you detect the immune response indicative of sepsis, and then you start that blood culturing mechanism, that blood culturing technique earlier than you would if you wait for symptoms to occur, right? So let's say the immune response starts to occur maybe 24 to 48 hours before sepsis symptoms show up. So if you can start the process of identifying the bacteria 24 or 48 hours sooner than you normally would, that just saves that time. And it's a race against time with sepsis. So anything you can save time-wise is just going to benefit the patient.
Gotcha.
So,
Big game changer there.
right. So, but then in a viral infection, you're going to rapidly show symptoms. And there are no antibiotics for viruses. All you really do is, you can give antivirals, for example, to slow the virus down and give your immune system time to get on top of it. But ultimately, you're going to rely on your own immune system to take care of a viral infection, because there are antiviral treatments, of course, to slow the virus down, but there's no way to kill a virus specifically at this time.
Yeah, just amazing to me. The human body can mount that immune response, which of course generally allows us to remain living and not die from it. But as you're describing, it sometimes needs to take the human host to the point of death to kill what is killing that human, and sometimes it just overshoots.
Yeah. The immune system is an amazing system, but it can often work against us as well.
Yeah, interesting. As the title of the podcast states, your current main project there, and I forgot to mention, you're at the Georgia Institute of Technology, or what's the precise school name there? Research.
The Georgia Tech Research Institute.
Georgia Tech Research Institute.
Yeah,
and
so
the main project, sorry?
No, Georgia Tech is, obviously the school, academic school. It's right here, and then Georgia Tech Research Institute is sort of the rather than less academic and more applied research.
Gotcha.
But we're all under the same roof.
So it just works with the same way as a lot of universities and research arms?
Yes.
Okay, got it. Thank you. And your current main project you're studying is how a common probiotic bacteria might be able to boost our immune response to influenza. Could you please describe the project you're working on?
Yeah. So influenza vaccines are generally given, intramuscularly in your arm. Oh, and they can also be administered intranasally. Have you ever had an intranasal flu vaccine?
I don't think so. No.
Yeah, it's just, they squirt a fluid up your nose instead of, sticking it in your arm. So there's two components to your immune system. There's the internal systemic immune system, and that's what's activated when you get an intramuscular vaccine. But another very important part of the immune system is kind of the externally environmentally facing part of your immune system. That's the mucosal immune system, your gut lining, your respiratory tract. As I said, the systemic immune system is activated by an intramuscular vaccine, and the intranasal vaccine is better at stimulating the mucosal immune system. I think when they compare them side to side, the intramuscular vaccine is more effective than the mucosal vaccine when taken separately. But I think ideally you'd have a combination of the two. But that gets very expensive to administer two vaccines for a single disease. So I don't think that's I don't think that's practical. So they seem to be sticking with the intramuscular vaccine, but if we could develop an adjuvant to that that can also stimulate the mucosal immune system, I think that would be ideal. So the idea here is to, a vaccination is simply presenting the immune system with a, a killed virus so that it can see the structure, recognize it, and build antibodies against it. So in this systemic immune system, we're talking about IgG antibodies and IgM antibodies. And in the mucosal immune system, it's more about IgA antibodies. And they all basically do the same thing, recognizing the pathogen, but they just stimulate different parts of the immune system when they find their target. So our idea is to modify a probiotic bacteria that would express an antigenic part of the virus and train the mucosal immune system to recognize that, so that when you do get an actual flu virus in your respiratory system, those IgA antibodies are waiting and they can deactivate the virus before it can cause an infection. So the probiotic bacteria we're going to be using is E. coli. E. coli is a very common component of everybody's microbiome, generally comprises less than 1% of the microbiome. Their functions are to sort of maintain population diversity, keep bad bacteria down, good bacteria healthy, working in cooperation with all the other microbes that are in there. They help digest food. Some strains of E. coli can create vitamin A, for example. So, they're beneficial host commensal probiotic bacteria. There are E. coli that are, of course, quite hazardous. You hear about E. coli foodborne diseases breaking out from contaminated foods. E. coli 0157H7, for example. The difference between these pathogenic E. coli and the good commensal E. coli is that the pathogenic E. coli code for and express a toxin, Shiga toxin. And that's what causes the foodborne disease. This toxic makes you very ill. But the bacteria that we would be using do not have Shiga toxin. They're very harmless commensal bacteria that are a normal part of your gut flora.
So it's just the delivery vehicle.
Yes, exactly. So the idea would be so bacteria have an outer membrane that's comprised of various protein components. And we'll take one of these components, and what we will do is we will fuse a portion of the hemagglutinin receptor from the influenza virus. We'll fuse that with that bacterial coat membrane protein, so that when the bacteria replicates, a portion of that outer coat membrane protein will have a little chunk of the influenza virus on it. And that will present that to the immune system. The immune system will recognize that and develop antibodies for that specific influenza structure. And then when the actual virus shows up, the immune system is trained to see that hemagglutinin epitope and attack that virus.
And I know you're using CRISPR, which is normally sort of gene editing, but is this actually editing the gene of the E. coli or not, just because it's on the cell membrane?
Yeah, so the use of we're actually not planning to use CRISPR. We may use it if the current approach fails, but CRISPR is just one of many genetic tools that as you say, alter the genome of what you're trying to work with. So yeah, you would go into the E. coli genome, you would find the gene that codes for that outer membrane structure protein, and then you would insert in there that viral hemagglutinin fragment, so that when that protein's made, it also makes a portion of it that's the viral structure as well.
Yeah. That is it's so amazing to me. I get the general concept, but it's still like a black box. Oh yeah, we just go in and snip this out and put in the right
Yeah,
piece.
yeah, it's extremely complicated
Mm.
molecular gymnastics,
Mm.
step by step by step, building from what you previously did to ultimately uh, change a genome in a bacteria in such a way that uh, the bacteria can still carry out its normal functions, but also have this extra uh, piece of protein that sticks on its outer coat that is different from what it would naturally express.
Probably like a lot of research, you have a collection of failures until you get some breakthroughs and build upon that, essentially.
Oh yeah, I mean, uh, all of the stuff that we're trying to do has been done before. We're not really inventing any specific technique. We're just doing it in slightly different ways to get a very specific outcome.
For the
So yeah,
purpose.
I don't want to sound like I'm taking credit for developing any of these techniques. I'm standing on the shoulder of giants.
Sure. Yeah, as anybody does in research field, no doubt. Yeah. But to be able to, the end goal is to lessen the severity and deaths of the influenza virus.
Yeah, absolutely. And it's a very tricky thing developing seasonal influenza vaccines. They have to figure out which strains are circulating out there and rapidly manufacture up a vaccine. And vaccines today will have two, three, or even four different strains that they're targeting in a single vaccine. Um, so it's a very delicate system, and a rapid system that we use every year to try to figure out what influenza strains are going to be the threat this year, and developing the proper vaccines for it. Uh, so having a platform like a probiotic that you could rapidly change uh, could be very cost effective. Probiotics are very cheap to make uh, as compared to making a vaccine. So uh, the point of doing this uh, is to provide an adjuvant to the actual seasonal influenza vaccine, but something that's very low cost. You don't want to, you know, as I said earlier, we don't give an intramuscular vaccine and a nasal vaccine because it's cost prohibitive. Um, so if we're going to do anything in addition to the seasonal influenza vaccine, it has to be very low cost.
So if it's so low cost and easy and supermarket shelves are filled with probiotics, why can't I just, if this works out and it passes human trials, et cetera, why can't I just pop this probiotic and not worry about a flu vaccine?
Uh, that's a good question. I mean, maybe you could someday, but uh, this concept, even if it worked, would have a long way to go to replace what we're doing now for uh, annual influenza vaccines. Uh, so I wouldn't want to make a prediction like that.
Hmm. But they're ultimately meant to team together, probiotics and the vaccines.
In the current concept, yes, absolutely.
Gotcha, gotcha. And you've been at this your team has been at it for about how long?
A little less than a year.
Okay.
We're very early stages still. We're trying to determine, uh, you know, which part of the hemagglutinin protein that we want to express on the outer bacterial coat uh, membrane, and lining up all of the different recombinant DNA platforms that we'll need to use to actually create the genetic change that we want to institute. So still early stages.
Imagine, it sounds to me like building a house or a super highway. You've got multiple steps and processes that you're working through. So it sounds very, very complex.
It is. It is.
Yeah. And I read an article about this project, and it stated that the bacteria would be modified so they could not reproduce, preventing them from becoming part of the body's gut microbiome. So in other words, a permanent member of the gut microbiome. Why is that step so important?
Yeah, that's a great question. I think our feeling is that, as with any vaccine, you get your vaccine shot, the immune system recognizes it, but ultimately, that that concoction is absorbed and degraded and goes away. Uh, you don't you, you don't want to overstimulate the immune system, because, as we've discussed, that can be catastrophic. Um, so we don't want to put anything that's going to constitutively express the influenza target protein. Um, Number one, we don't know what that would do. If you continuously express it, uh, maybe the body starts to accept it as being normal, and you no longer develop
Mm,
antibodies against it.
mm.
Um, so that it seems prudent to uh, not allow this bacteria to replicate and maybe become a permanent resident in the gut, but rather just let it simply pass through. Uh, and then in that way, you can control the amount of bacteria that you're delivering through just dosing.
Yeah, interesting. Making a non-reproducing bacteria. That's great. Yeah, in regard to vaccines, and there's everybody knows there's been a lot of controversy about COVID vaccines and lots of talk about gain of research, gain of function research rather. And so if you're talking about editing, parts of a bacteria, should anybody be concerned? Is this the same as gain of function research?
No, I would say no. It's maybe a gain of a structure,
Hmm.
but there's no functionality to that. Um, and in fact, if we take the further step of making sure this bacteria can't replicate, that's really a loss of function.
Hmm.
Uh, so no, there's no gain of function uh, per se in this project.
Yeah, that makes sense. A loss of function. That's exactly correct. It's sort of you built in these because they can't reproduce, sort of a self-destruct button in it.
Yes, and some of your viewers may or listeners may wonder, well, if it can't reproduce, how can you grow it in the first place? The trick there is to grow it, is to provide that missing component in trans while you're growing it up. But then after the bacteria is on its own, it will not have that uh, that supplied uh, element that it needs to reproduce.
Interesting. That question had not occurred to me, but so thank you for that explanation.
Sure.
What you were talking about something very specific, the project you've been working on, but in general, what really excites you about the human microbiome?
Oh wow. Uh, it there's we have just scratched the surface of what we're going to be able to do with the human microbiome. It's probably it's going to revolutionize health completely. Um, there's so much that has to be done before we get to that point, and it's very complicated. Everybody's gut microbiome is different. There are hundreds of different species, and they're different from person to person. The dominant populations can be different from person to person. Your diet, your environment, your genetics, all of these things interplay with how your microbiome is comprised. And with all of that complexity from individual to individual, it's really hard to find out which population is doing specifically what, because I believe there's a lot of redundancy in the microbiome.
Hmm.
I think if a certain bacterial population is fulfilling a very specific niche in one person, well, a different population can provide that same niche function in another person. So there's a lot of bacteria that can be completely are completely redundant. They do the same function in one person than and another population does in another person. Uh, so that sort of has to be parsed out. What are the main core functions that many different bacteria are doing, but you can distill it down to what is that core function. So figuring out exactly what roles the bacteria are playing, a lot of that is known, but a lot of that is still unknown. So just kind of figuring that out is very interesting, and it's very hard to do because there aren't any really good controls out there. Everybody is so different. It's hard to have a control population in doing some of these experiments. But once we do figure that out, the real exciting part is going to be, how do we alter the gut microbiome for the better? How do we target a very specific population for depletion, elimination, and then how do we introduce a bacteria that we do want without upsetting the whole balance? So I think that's where the really exciting part of the microbiome is. I take it for granted that it's going to be a huge seismic shift in healthcare, is managing people's gut microbiome. Exactly mechanistically how you do that is going to be also extremely fascinating. I see phage bacteria becoming a very key component. Phage bacteria attack and kill very specific species and strains of bacteria. And so you could grow up a whole cohort of all of these different phages, and you could apply them to eliminate very specific populations. And then through probiotics, deliver the replacement bacteria that you want in their stead. That's also going to be very fascinating. A lot of your general probiotics that you take in the morning, they're all gram-positive bacteria that can be easily grown and lyophilized and put into a pill. That's inherent in their physical structure. But there are a lot of bacteria that are gram-negative that don't have that capability. You can't desiccate them and dry them and deliver them in a probiotic form. So figuring out mechanisms to grow these other types of bacteria that are equally as important to your gut and delivering them so that it gets through the stomach and gets to where they're supposed to be, that's also going to be very fascinating work.
Like so much research, and maybe you've seen this, been frustrated by it at times. There seems to be a large gap of time between valid research and clinical practice. And it seems to me that the average physician, clinician nowadays, although a lot of growth in understanding, the average person is clueless. If you gave them a shotgun metagenomic stool test, they wouldn't know what to do with it. So that's a challenge.
Yeah, absolutely. So yeah, hand in hand with all the rapid changes that are going to be taking place centered around managing the gut microbiome is the education component.
You mentioned in one of your notes an example of a fecal transplantation, or FMT, and that you take fecal material from a donor and put it in a recipient. And specifically, you mentioned somebody with Clostridium difficile, C. diff infection, which causes thousands or tens of thousands of deaths a year. And I first discovered this. This is how I got into gut health in general. I don't know, 10 or more years ago, I happened upon this FMT thing, and I saw it takes away the C. diff within 24 hours, and my mind was blown. And I thought, this is the game changer, right? And I remember telling Tim and your brother had long conversations about this. And that's when he says, oh yeah, my brother works for the CDC, et cetera, et cetera. But that's how my interest got started, but just so powerful, as you say.
Yeah, yeah. As a proof of concept, that really kind of stands alone
Hmm.
as evidence. Uh, yeah, C. diff is a can be a normal part of somebody's gut microflora, but it's kept at a very low level because of all the other populations that are doing their own thing and crowding it out. Uh, what what niche C. diff may fill as a very minor component, I have no idea. Uh, but it's when you take for example, an antimicrobial course of treatment and you kill off a lot of your gut bacteria, C. diff has a chance to expand, and once it expands, the other bacteria can't keep it down. So a fecal microbiome transplant just simply restores that balance. It keeps that C. diff down to the very low level that it's supposed to be at. So yeah, very clear cut treatment and a very clear cut reason why it works, and just directly points to uh, how powerful manipulating the gut microbiome can be for human health.
Hmm. I've
I
done
mean,
some. Sorry.
oh no, I mean, you get beyond C. diff and you start getting to, you know, colitis and irritable bowel syndrome.
Mm-hmm.
These are diseases that are uh, should be very amenable to controlling through manipulation of the gut microbiome. Uh, there may be other things going on, but that could certainly contribute to uh, alleviating some of the symptoms and pain that these diseases cause people.
Yeah. Yeah, I totally agree. I've done a few podcasts and I create some video content for this one YouTube channel. And there were these two infants and they had identical outcomes where both of them had extreme high levels of Bacteroides fragilis, 50, 60%, and little to no bifidobacteria. And because they're infants and they're getting HMO through the breast milk, they were given a specific strain of B. infantis, Bifidobacteria infantis, and the infantis skyrocketed and the Bacteroides fragilis decreased, and they were in direct proportion to each other. I mean, you can see the chart. One goes up, the other goes down. So that positive competition is so vital.
Yeah, absolutely, and yeah, every bacteria that's there may be a lot of freeloaders in there. We're living off the other bacteria, but uh, yeah, it's amazing how we have evolved to use this internal infection, to call it to call it what it is, and use it to our advantage. A symbiotic relationship with these bacteria. Uh, that's really amazing.
Yeah. Yeah, and we both are equally amazed. My perspective, I mentioned, I'm a pastor. That's how we met. So my position would be these were divinely designed, put in there. But I understand, the evolutionary process have that viewpoint as well. But that's my
I,
personal conviction on the matter.
I think there's room for both of those viewpoints.
Sure, sure. Yeah. You also mentioned the it's so complex you want to benefit it without harming it. For example, I've done I don't know probably 6 or 7 stool tests on myself the last 4 years, and I had no bifidobacteria. I had as much as 8.5%, which is crazy high for an adult, and generally, would be a good thing. Then I did some other things and my bifido crashed and I don't know why. So I'm a living example of trying to mess around maybe too much.
That's interesting. I don't do that myself, so I don't have anything to compare with directly. But I think you would be surprised how much the microbiome changes day to day, week to week, depending on what your diet is.
Mm,
Uh, so if you're on a steady diet, doing the same thing. Your gut microbiome should be relatively static. So that's interesting that you had that experience.
Uh, Mike, this is sort of I say wave the magic wand sort of question. If you had unlimited funding and resources, what would you love to study?
That is a great question. I think, I guess I would, rather than, there's so many people studying the gut microbiome right now. Uh, I would not, I would not want to compete with those folks doing great work. But there are other components of the gut microbiome that don't get enough attention, and you know, the fungome, the fungal component of the gut microbiome, I think is probably also extremely important. And we just don't really know what contributions they're making. Uh, I think that would be fascinating to study. There are also viruses that play roles in the gut microbiome. Um, there are plenty of human enteric viruses, rotavirus, for example, that you hear cruise ships, so there are a number of enteric viruses that cause human disease that may also uh, have a very uh, small or large role in the gut microbiome. Have no idea.
Hmm.
Uh, I think that would be interesting to study. Uh, so yeah, going off the beaten path, looking at other aspects of the gut microbiome besides just the bacteria, I think would probably be very interesting, understudied, and maybe could provide some useful insights into overall gut microbiome function and maintenance for health.
And
So yeah, go off the beaten path and look
mm,
at things that other people may not be looking at.
and sticking to your sort of your original love, viruses especially.
Yeah, there you go. And yeah,
Hmm.
absolutely. And bacteriophages or viruses, they're just
Mm.
viruses for that target bacteria instead of mammalian cells, right? So that's a, a whole nother, I mostly work with mammalian viruses because they cause human disease, but viruses that target bacteria are also uh, fascinating.
Mm-hmm. Hey, I have a very, very practical question for you. You mentioned rotavirus. Last week, my son and his whole family were extremely ill. It wasn't one of these 24-hour deals, and it continued on with gut symptoms, and then, yeah, it was really, really wicked. So it made me think, what? So to kill a virus, since it's hijacking your own cells, is it ultimately the immune response that kills the virus, or how does it finally go away?
Yeah, it is the immune response that finally eliminates viruses from your system. That's exactly right. Uh, if you haven't been previously exposed to that virus, and you don't have uh, you know antibodies waiting in the wings, just hanging out, ready to attach to a virus when it shows up, your body has to develop them. Uh, so it goes through the whole process of uh, you know being an infected cell is digested by a macrophage and shows those foreign proteins on its surface, and those are recognized by B cells, and then they make antibodies against that epithelium. It's a very long process that your immune system takes to develop antibodies for a new pathogen that it's never seen before. But yeah, ultimately, it is your immune system that gets rid of viruses. We don't have magic antiviral bullets for the most part for viruses like we do for bacteria.
Hmm. So this particular rotavirus was obviously new to my family, and perhaps quote unquote worse, you know,
Well,
in some regards.
yeah, not necessarily. I mean,
Okay.
I mean, maybe it had been exposed before, and if your immune system wasn't already pre-primed, their illness may have been much worse than it actually was,
Sure.
right?
Yeah.
Yeah.
I guess that's always the case, isn't it? Yeah. Interesting. Well, Mike, is there, we covered a lot of topics. Is there anything else you wanted to share? Anything about current research, past research, interests?
I would just save that for podcast 2.
Hey, that sounds good. That sounds good. I got you on tape here, committing to that someday.
Yeah,
So
no, it's been great talking to you, Rich, and I appreciate you letting me part of your podcast. I love talking about science, and it's a passion of mine.
yeah, fantastic. Well, thanks again so much, Mike, and take care.
Take care yourself, Rich.
All right. Yep. So long.